This is Fencer Wang speaking. I'm a physician of Chang'an Clinic of Taiwan, Kaohsiung. Today, what I want to speak to you is we can eat protein in early stage of acute virus infection. This conception is still immature currently, and now I'll tell you why. Survival of the fittest is the main principle of evolution. Nowadays, we can see that almost all animals become anoretic when they get an infection. Anoretic here means don't like to eat food. It is not a virus, but our immune system that makes you anoretic when you get a virus infection. Immune cells secrete cytokines to inhibit the appetite. Finally, we can see that infected animals which do not want to eat survive the disease and exist after the long-term evolution. But where are the animals whose appetite don't change or become well when they get an infection? I think they are all eliminated from evolution. I would like to tell you first, the most effective method to overcome an acute virus infection is fast. However, it's difficult to endure hunger if one is not severely ill. So the compromised method is that inhibit the protein and lower calories intake in early stage of acute virus infection. This picture shows simple profile of virus. A virus has internal nucleic acid, DNA or RNA, and an outer capsid composed of protein. Some viruses have an envelope, which is lipid bilayer, outside the capsid. Though every part of a virus has its importance, the DNA or RNA is the most important for virus. DNA or RNA is main component of virus. Purine and pyrimidine are main components of DNA and RNA. Amino acids are main components of purine and pyrimidine. Amino acids are almost the whole components of protein. We can get a relationship. Protein can be decomposed to amino acids. Amino acids can be used to synthesize purine and pyrimidine. Purine and pyrimidine can be used to synthesize DNA and RNA. DNA and RNA can be used to assemble virus. Theoretically, if there is no protein, there is no virus. But protein is an important nutrient for body. We can live without protein. Immune system is skilled in the tactic of this relationship. When infected by viruses, immune system inhibits nutrition intake and thus inhibits the synthesis of purine and pyrimidine. There are two ways for synthesis of purine and pyrimidine. 1. Salvage pathway. 2. De novo pathway. Salvage pathways of purine and pyrimidine, they exist in each cell or body. It's the way to recycle purine and pyrimidine to make conversion between different derivatives possible and to fit requirement of different biochemical reactions. De novo pathways of purine and pyrimidine. De novo means from the beginning. It's the way to synthesize purine and pyrimidine from the beginning. Amino acids and other materials are incorporated into purine and pyrimidine rings step by step. Liver cells and immune cells have the ability to de novo synthesize purine and pyrimidine. But common or infected cells can do this. They rely on the supply from liver. De novo synthesis will proceed to satisfy the requirement of large amount of purine and pyrimidine, such as cell reproduction and virus replication. Immune system inhibits the synthesis of purine and pyrimidine and then inhibits the reproduction of virus. Principle of immune reaction. Inhibit the reproduction of virus and use as little immune strength as possible to demolish the virus. The immune reactions are also harmful to human body. Sometimes people want to increase the immune strength when they get a virus infection. It seems not correct, for immune reactions are also harmful to human body. The very extreme case is immune storm, which means too strong immune reactions damage our tissue and endanger our life. We don't clearly understand why some people completely recover, but others are dead from the same virus disease. The latter may be ingest too much protein in the early stage of acute virus infection. It makes massive replication of virus and results in severe tissue damage and has more possibility to become secondary bacterial infection. Strategy of immune reactions to virus is to control the energy and the nutrients, and then control the synthesis of purine and pyrimidine, and finally control the replication of virus. How come? 1. Inhibit ingestion of nutrients. In fact, the calories intake is also inhibited in the meanwhile. 2. Increase internal energy expenditure while energy intake from outside is poor. 3. Block the use of backup energy, lipid. 4. Decomposition of muscle protein maintains serum amino acid level and becomes almost the only nutrient and energy source. The purpose of above procedures is the following point. 5 amino acids absorbed by liver cells are there to synthesize glucose, triglyceride, and acute phase proteins. Hence, very few purines and pyrimidines can be synthesized. Let's think again. Amino acids absorbed by liver cells can be used to synthesize glucose, triglyceride, acute phase proteins, purine, and pyrimidine. Block ingestion of outside energy, and block usage of backup energy, lipid, and increase energy expenditure of body. Now glucose is largely exhausted, and the only replenisher is protein. Most amino acids absorbed by liver cells are led to synthesize glucose. The role of synthesis of triglyceride is incredible. Normally, the excessive ingested nutrients are there to synthesize triglycerides. They are transported to and are stored in adipose tissue. But in acute virus infection, the nutrients intake is poor. While liver cells reversely use amino acids to synthesize triglyceride, when nutrients intake is poor and its usage is blocked. Its purpose is also to exhaust the amino acids absorbed by liver cells and ensure these amino acids not to synthesize purine and pyrimidine. 6. Immune cells can perform de novo synthesis of purine and pyrimidine. They can reproduce and function normally. Let's review important immune events. 1. The digested and absorbed purine and pyrimidine bases and their derivatives are metabolized by epithelial cells of intestine and are expelled through urine. Only about 5% of purine and pyrimidine contained in the food can enter the circulation and be used. We should think here, purine and pyrimidine are such important materials we need, and they are plenty in foods. It takes many efforts to digest, absorb, metabolize, expel, and synthesize them. It may also need a long period of time to evolve each step. The step of synthesis even needs many molecules of ATP. Why don't our bodies just directly use them that are digested and absorbed? So it must imply critical evolutionary meaning. 2. Immune system controls the de novo synthesis of purine and pyrimidine. It is performed mainly in the liver. Most cells do not have the ability to do it. 3. Inhibited ingestion of nutrients, especially protein. 4. Decompose muscle protein to maintain the serum level of amino acids. 5. Inhibit lysis of fat to fatty acid in adipose tissues and inhibit formation of ketone bodies in liver. 6. Inhibit LPL, lipoprotein lipase, which can decompose triglyceride of lipoprotein to fatty acid, and the triglyceride synthesized from liver can't be energy source either. 7. Increase of energy expenditure, especially when fever, forces the amino acids absorbed by liver cells to synthesize glucose, which is the main energy source in the acute stage of virus infection. 8. Some amino acids absorbed by liver cells are used to synthesize triglyceride, which seems useless and luxurious, for the LPL is inhibited and the nutrients and the calories intake are poor. Its purpose is to exhaust the amino acids in liver cells. 9. Some amino acids absorbed by liver cells are used to synthesize acute phase proteins, which are thought to be helpful to immune reactions. 10. No or very few amino acids in liver can be used to synthesize purine and pyrimidine, so DNA and RNA can't be synthesized and the virus can't replicate. 11. Immune cells can perform de novo synthesis of purine and pyrimidine, so they can normally reproduce, function, and produce antibodies. Under the effect of cytokines and stress hormones, there is no or little nutrients intake, and the muscle proteins are decomposed to maintain the serum amino acids level. The immune systems can normally get amino acids from serum for cell reproduction, antibodies production, and cytokine secretion. Because immune cells can perform de novo synthesis of purine and pyrimidine, they need not rely on the supply of liver. Amino acids absorbed by liver cells are used to synthesize glucose, acute phase proteins, and triglyceride. The acute phase proteins are thought to be helpful to immune reactions. However, the synthesis of triglyceride is very bizarre, for the ingestion of nutrition is poor and its usage is inhibited. It is unwise to think this step as an evolutionary defect. In fact, it should be part of immune strategy. Exhausted amino acid to synthesize triglyceride can prevent synthesis of purine and pyrimidine. The most effective process to exhaust amino acids absorbed by liver cells is the synthesis of glucose. It is largely produced even more than it is needed. Serum glucose level is elevated, and it stimulates the secretion of insulin. Insulin inhibits the lysis of lipids. Therefore, the fatty acids and the ketone bodies can be produced, so they can be energy source as in starvation. Glucose, more correctly amino acids or proteins, becomes the main energy source in the acute stage of infection. Fever further increases its requirement. Amino acids obtained by liver cells are massively led to synthesize glucose, acute phase proteins, and triglyceride. So, purine and pyrimidine are hard to be produced. Formation of DNA and RNA is limited, and the virus cannot replicate. In acute stage of infection after feeding, large amounts of glucose, amino acids, and fatty acids are ingested. The ingested glucose and fatty acids may reduce the amino acids absorbed by liver cells to synthesize glucose and triglyceride. So, there are more amino acids can be used to perform the de novo synthesis of purine and pyrimidine. In addition, there are small amounts of purine and pyrimidine can be absorbed from food. Therefore, DNA and RNA synthesis can proceed, and the virus can replicate, and the disease will be worse. Definition of acute stage of virus infection: 1. Initial 4 days. 2. Any time when body temperature elevates. 3. Any time when general soreness, malaise, sleepiness, or poor appetite appears without evidence of any other contributing factor. The definition is basically according to clinical finding and trial. Any time means any time during this infection. In other words, if the infection disease enters the 5th day and there are no acute symptoms such as fever, general soreness, malaise, weakness, and sleepiness, it is not in the acute stage. Treatment principle in acute stage of virus infection: 1. No protein should be ingested. 2. The amount of ingested calories, purines, and pyrimidines must be limited. 3. In severely fatal virus infection, nothing should be ingested except water, electrolytes, and vitamins. Treatment in acute stage of virus infection: 1. If there is no elevation of body temperature, inhibit protein ingestion and lower calories intake to 80% of normal daily requirement for 48 hours. Normally, the basic daily calories requirement for a adult is about 1,600 to 2,000 kilocalories. It varies with sex, body weight, lifestyle, and so on. And 80% of normal daily requirement is about 1,280 to 1,600 kilocalories. You'd better go convenience store and buy packaged food. You can get the protein and the calories data in the ingredients. If you can calculate calories precisely, don't worry, just don't be full and keep mildly hungry. That's okay. 2. If there is mild to moderate elevation of body temperature, 37.6 Celsius to 38.5 Celsius, inhibit protein ingestion and lower calories intake to 50% of normal daily requirement at the first 24 to 36 hours. Then continue to inhibit protein ingestion and lower calories intake to 80% of normal daily requirement the following 36 to 48 hours. Total time of food control is 72 hours. The 50% of basic daily calories requirement is about 800 to 1000 kilocalories. Again, if you can't calculate it precisely, just keep moderate hungry all day. 3. If body temperature is over 38.5 Celsius, inhibit both protein and calories intake at the first 24 to 36 hours. Then continue to inhibit protein ingestion and lower the calories intake to 80% of normal daily requirement the following 36 to 48 hours. Total time of food control is 72 hours. No medication is needed. Immune system alone with diet control can overcome even the acute fatal virus infection. However, combined treatment with antiviral medicine may be helpful. Chicken, duck, fish, pork, beef, egg, milk, bean, bread, noodle, rice, porridge, wheat, grains of all kinds are all protein-containing foods. They must be avoided when food control. No protein foods would be better to be leaves or vegetables, not to flavor with MSG or soy sauce, fruits without their seeds, and packaged foods of which ingredients mark protein zero. The contents of natural foods are complex. It's better to go to convenience store to buy packaged foods whose amount of protein and calories are clear. No protein and low calories foods would be better to be diluted juices without a dress, water with light salt and sugar, packaged foods with no protein and low calories, and so on. 1. Food control has to start as early as acute virus infection is considered. If it does not start at the very beginning, the infection may last 4 more days. 2. There is no need to control the protein and calories intake when it is not in the acute stage. 3. Any ingestion should expel high purine and pyrimidine foods. Not in the acute stage means the infection disease enters the 5th stage and there are no acute symptoms such as fever, general soreness, malaise, weakness, and sleepiness. High purine and pyrimidine foods usually accompany with high proteins and high calories, except asparagus, mushrooms, bean seedlings, bean sprouts, liver, and so on. They should be avoided too, because there are 5% of purines and pyrimidines that can enter the circulation and may be used by virus. There is no clinical symptom in the incubation period. The amount of virus is low and only trivial innate immune responses happen. If the virus amount increases over the critical value, the disease happens. The immune system is formally activated. It's the day 1. The infected cells can produce interferon alpha and interferon beta to protect the neighboring cells. Generally, NK cells are the main power to kill the infected cells in the initial 2 days. Another important cell of the innate immune system is a macrophage. Macrophages at the infection site can phagocytose virus and virus-infected cells, and they can kill virus-infected cells. On the 3rd day, effector CD8 plus T cells, CTLS, which are more effective to kill the infected cells, take part in this combat. It means the adaptive immune system begins to participate in the combat with virus. On the 4th day, the amount of the NK cells decline, and the CD8 plus T cells, CTLS, rise. The CTLS take over the position of NK cells to continue to kill the infected cells. On the 5th day, serum IgMs against the virus appear. The IgMs circulate in blood vessels and prevent viruses spreading from one side to another. Unless the amount of viruses is too large, these IgMs can restrict the viruses in a local site. In clinical practice, we can see symptoms of virus infection begin to remit. On the 7th day, serum IgGs against the virus appear. The IgGs can penetrate blood vessels to eradicate the viruses that are scattered in different tissues. Theoretically, the progressive speed of immune responses is near constant, but the strength is different. Actually, virus antigen is the ultimate driving force of immune response, of which strength is determined by the amount of virus antigens. The replication rate of virus varies very much. If nutrition supply is sufficient, the virus will replicate to a very large amount, and it will induce a series of bigger immune responses, which will make the disease severer. If one receives the protein-calorie control in the very beginning of virus infection, the amount of virus is low and the replication of virus is limited, but the reproduction of NK cells is near normal. After 48 hours, the amount of virus is still low, but the NK cells reproduce to a larger amount. Besides, CTLS, which are specific and powerful killers to virus, begin to appear. The immune strength is further enhanced. If one receives normal diet at the moment, virus will have no chance to overcome the immune system. If a disease accompanies the fever, the virus is supposed to be more virulent, and we take 72 hours protein-calorie control to let the immune system become more developed and powerful. Again, the virus will have no chance to aggravate the disease. On the 5th day, the IgMs appear. They circulate in the blood and prevent viruses from spreading via it. At the moment, the immune system is well reinforced, and there is no need to control the foods unless the amount of viruses is very large, and they can break through the defense of IgMs and spread to other tissues. If it occurs, the acute symptoms such as fever, general malaise, general soreness, poor appetite, and sleepiness will remain, and the protein calories should be controlled till the acute symptoms subside. Acute symptoms are caused by cytokines, which are mainly secreted by immune cells. If the amount of viruses is large, more immune responses are induced, and more cytokines are secreted, and more severe disease can be noted.